Hi, this is Alex from devstories.eu. Scanpy, Single Cell Analysis, Episode 6 of 16. Your clustering algorithm just split identical T cells into two distinct groups. They are the exact same cell type, but the algorithm separated them simply because one group happens to be actively dividing while the other is resting. To fix this, we use cell cycle scoring and regression. Cell cycle heterogeneity is a massive source of variance in single-cell data. If left unchecked, the highly expressed genes driving mitosis will overpower the subtle gene signatures that define actual cell types. You end up with clusters defined by a temporary state rather than true biological identity. To solve this, Scanpy provides a dedicated function to score genes based on the cell cycle. You pass this function your single-cell dataset along with two specific lists of known marker genes. One list contains genes that are active during the S phase, the synthesis phase of the cell cycle. The other list contains genes active during the G2M phase, the mitosis phase of the cell cycle. The scoring function evaluates every single cell and calculates two continuous metrics, an S score and a G2M score. It does this by looking at how strongly those specific phase genes are expressed compared to the background expression level of the cell. Based on these two scores, the function also assigns a categorical phase label to each cell in your metadata, labeling it as S, G2M, or G1 if neither score is particularly high. Now that you have quantified this effect, you need to scrub its influence from the dataset. This is where you use regress out. You instruct regress out to look at the S score and G2M score columns that were just added to your cell metadata. The algorithm then builds a linear model for the expression of every single gene across all cells using those two cell cycle scores as the predictor variables. It calculates the residual, which is the exact amount of gene expression that cannot be explained by the cell's position in the cell cycle. This residual value becomes the new corrected expression level in your dataset. Here is the key insight. People often confuse regression with batch correction. They are related concepts, but they are fundamentally different tools. Batch correction methods are designed to align discrete categorical groups, like samples collected on different days or sequenced on different machines. Regress out is specifically designed for continuous confounding variables. You use it for continuous numerical gradients like these cell cycle scores, total counts per cell, or the percentage of mitochondrial genes. It models a mathematical slope and flattens it. Once you run this regression step, the heavy biological bias of cell division is mathematically removed from the expression matrix. When you run your clustering algorithm again on this corrected data, those dividing T cells and resting T cells will snap back together into a single cohesive cluster. The algorithm is no longer distracted by the noise of DNA replication. Regressing out cell cycle scores ensures your downstream analysis clusters cells strictly by what they are, rather than what they just happen to be doing at the moment they were sequenced. That is all for this one. Thanks for listening and keep building.